today we're going to talk about and carry out a lab on the topic of Kas and equilibrium.
As you should know, Bronsted-Lowry acids are proton donors and Bronsted-Lowry bases are proton receivers. However, just because something is an acid doesn't mean it will donate a proton 100% of the time.
The process of donating a proton is called dissociation, and methods of measuring dissociation include percent ionization and the acid dissociation constant, Ka, which is the most important factor. The definition of Ka is the concentration of H plus times the concentration of the conjugate base over the concentration of the acid.
Ka is useful because it is information that can be used to calculate what the concentrations of H plus, conjugate base, or A minus, and the initial acid, HA, are at equilibrium. We can calculate all of these in the system. I screwed that up, didn't I?
Just keep going. We can cut that out.
Ka is useful because it is information that can be used to calculate what the concentration of H plus, conjugate base, or A minus, and the initial acid, or HA. This means that when we know the concentration of an acid and that acid's Ka, we can calculate the concentration of all three components in the system at equilibrium. It also means that we can predict changes in concentration of acidic systems.
Only weak acids have a Ka because they do not fully dissociate.
In a weak acid, the Ka of the acid determines to what extent the acid will dissociate, because it will not fully dissociate. For this reason, Ka is used to measure the strength of an acid, because in a very weak acid, very few molecules will undergo dissociation, whereas in a strong acid, almost all molecules dissociate. Acid-base equilibrium is also affected by the same factors that affect other equilibrium systems according to Le Chatelier's principle. A change in concentration will cause the reaction to proceed in a certain direction in order to preserve equilibrium. We will try to investigate this later.
As is the case with other equilibrium constants, a change in temperature will affect the concentration of the products and reactants, but it will also affect the constant itself. Acetic acid dissociation is an endothermic process because energy is needed to break intramolecular bonds. This means that when the temperature is increased, the system will shift to the right, and when the temperature is decreased, the system will shift to the left.
The common ion effect is another important factor in equilibrium. Ka is calculated using the concentrations of species in the equilibrium system. But what if some of those species are already part of the solution? In that case, they will affect the extent to which the acid will dissociate. For example, if an acetic acid solution was created and acetate ions were already in the solution, the acetic acid would dissociate to an extent that was less than what it would otherwise. It is the equivalent of adding products to the system, which would cause it to shift to the left. In order to calculate the equilibrium concentrations of the species involved in the reaction, ice tables must be used in a manner similar to the process used to solve equilibrium problems. First, the initial concentrations of the species must be determined. In a simple problem, the concentrations of the products will be 0. Next, the concentrations of the products are solved for by creating an equation based on a known Ka.
We are now going to walk you through a lab that tests the concepts we just discussed. Pay close attention to the process because it will be important for understanding how to do the math that will be required after the lab is completed. After the lab, there will be analysis questions for you to answer.
These are some of the materials that are going to be used in the lab. One other important piece of equipment that is going to be used is a laptop with Logger Pro, which will allow us to measure the pH of the substances that are used in the lab. Part 1. Using a pipette to measure, we transferred 4.167 milliliters of a 6 molar acetic acid to a graduated cylinder. Then we rinsed a 50 milliliter volumetric flask with water 2 times and poured the 4.167 milliliters of 6 molar acetic acid into the volumetric flask. Then we filled to the line with distilled water to create a 50 milliliter solution of 0.5 molar acetic acid. We then rinsed the graduated cylinder with distilled water twice and dried it. Next, we transferred 2.083 milliliters of 6 molar acetic acid to a graduated cylinder to measure, and then transferred to a 50 milliliter volumetric flask. And then we filled the flask to the line with distilled water to create a 50 milliliter solution of 0.25 molar acetic acid. We rinsed the graduated cylinder with distilled water twice and allowed it to dry. To test pH, we opened up the pH preset program in Logger Pro from the school laptop, rinsed the pH meter with water twice, and then attached the pH meter to the laptop. We then poured the 50 milliliter solution of 0.5 molar acetic acid into one 100 milliliter beaker, and the 50 milliliter solution of 0.25 molar acetic acid into another. Both beakers were rinsed and dried beforehand. The pH meter was then submerged in the 0.5 molar acetic acid solution, and the pH of the solution was measured. We took out the pH meter and rinsed it. We then repeated the process for the 0.25 molar acetic acid solution and measured the pH of that solution. Here, we are measuring out the volume of acetic acid that will be necessary to create 50 milliliters of a 0.5 molar acetic acid solution. Getting precise measurements is important because even small amounts of acetic acid can have a large effect on the pH that is measured. The acetic acid was then added to a volumetric flask. Distilled water was then added to the volumetric flask to create a solution with a total volume of 50 milliliters. Again, precision was important, so we made sure we had the exact volume that was necessary. The pH meter was rinsed with distilled water to prevent impurities from affecting the measured pH. The pH meter was then connected to a computer that had Logger Pro in order for us to measure the pH of the solutions. Our first measurement was the pH of distilled water to ensure that our pH meter was working properly. To test, we poured distilled water into a beaker in order to be measured. To measure, the pH meter was added to the distilled water and allowed to sit for some time in order to get a precise measurement. The final measurement was around 7, which is exactly where water should be. The 50 milliliters of 0.5 molar acetic acid solution was then added to a beaker so that the pH could be measured. The pH meter was added and the solution was stirred and allowed to sit in order to get a precise measurement. Another 0.25 molar solution of acetic acid was created using more acetic acid. This was again added to a graduated cylinder. The acetic acid was then added to the volumetric flask and distilled water added in order to get the solution to 50 milliliters. During the process of adding distilled water, some bubbles formed in the volumetric flask, which could prevent the solution from being exactly 50 milliliters. In order to get rid of the bubbles, the volumetric flask was gently tapped on the table in order to get the bubbles to float to the surface. Again, getting 50 milliliters of solution was very important, so we used a pipette to get exactly 50 milliliters. Another beaker was rinsed and dried to prevent impurities, and then the acetic acid solution was added to the beaker. The beakers of acetic acid solutions were then labeled 0.25 molar and 0.5 molar in order to prevent confusion later on. The pH meter was rinsed between trials in order to ensure that the pH measurement was correct. The pH of the 0.25 molar solution was measured first. The pH meter was added to the beaker, and the solution was allowed to sit so that the pH measurement could settle on an accurate number. It took a fair amount of time in order for the pH measurement to settle, but mixing the solution with the pH meter did speed up the process a little bit. Next, the process was repeated for the 0.5 molar solution. The pH meter was added to the beaker, and then the solution was allowed to sit so that the pH measurement could settle. After an accurate measurement was recorded, the pH meter was rinsed in order to ensure that future tests would be accurate.
Part 2. Setting the 0.25 molar solution of acetic acid aside, we set up the hot plate. We placed a thermometer inside the 0.5 molar solution to measure and collect the room temperature. The solution was heated until it reached 75 degrees Celsius, and then we submerged the pH meter into the solution and collected the pH. The pH meter was then removed and rinsed. The solution was then allowed to cool down completely to room temperature.
Before measuring temperature, the thermometer was rinsed with distilled water in order to ensure it was clean before adding it to the solutions. Drying prevented excess water from changing the concentration of the solutions. The hot plate was plugged in and the solutions were added to the top in order to be heated. Room temperature was measured to be slightly above 20 degrees. The hot plate was then set to 95 degrees Celsius. This temperature was chosen because it is the boiling point of acetic acid. If any of the acetic acid were to boil out of the solution, the concentration of the solution would change and our pH measurement would change as a result. In order to ensure that our pH measurement was only a result of the change in temperature, the acetic acid solution was kept below the boiling point. The solutions were constantly watched during the boiling process for safety and also to ensure that the temperature did not rise above 75 degrees Celsius, which was our target temperature. When the temperature of the two solutions reached 75 degrees, the solutions were ready to be measured. 75 degrees was a high enough temperature in order to ensure that the difference in pH between room temperature and the tested temperature would result in a dramatically different pH. Again, the pH meter was rinsed. The pH of the 0.5 molar solution at 75 degrees Celsius was measured, and the results were drastically different from the solution at 25 degrees Celsius. The measurement was allowed to settle. The pH of the 0.25 molar solution at 75 degrees Celsius was measured next. Again, the same process of rinsing the pH meter was followed.
Part 3. We poured 4.015 grams of sodium acetate into the 50 ml solution of 0.5 molar acetic acid. We then mixed it with a stirring rod. When all the salt was dissolved, the pH meter was dipped into the solution to measure the pH.
In order to measure the correct amount of sodium acetate that would be added to the acetic acid solution, the balance was zeroed used with the weigh boat. A scoop was used to transfer sodium acetate from the bottle to the weigh boat. This was done slowly in order to make sure the changes in mass were incremental so that a precise measurement could be obtained. When the measurement was close to the desired mass, a sodium acetate was slowly removed from the weigh boat and then re-added in order to get the exact mass of sodium acetate that was desired. A stirrer was rinsed and then dried to prevent impurities from affecting the concentration of the solution. The sodium acetate was then transferred from the weigh boat to the acetic acid solution with care taken to ensure that all of the solid was removed from the weigh boat. The stirrer was then used to mix the solution in order to ensure that 100% of the sodium acetate was dissolved. We knew this was possible because sodium acetate is soluble. We stopped mixing the solution when solid was no longer visible. This meant that all of the sodium acetate had been dissolved into the solution. The pH meter was rinsed, and then the pH of the 0.5 molar acetic acid solution with additional sodium acetate was measured. The measurement was allowed to settle on a stable number.
Now that the lab is completed, go to the analysis and conclusion sections of the worksheet. They will ask you to perform math that is based on the lab we just performed. The conclusion questions will ask you to extend your thinking to other topics and require you to do research on the topics tested in the lab.